Okay, good morning, everyone. Good afternoon, good evening. This is Cherry Kubota. I'm a professor at the Ohio State University. Welcome to Indoor Ag Science Café February. I am so excited to have, I think this is a 60th Indoor Science Cafe, if I remember. So have been doing 5 years. Excellent. So just give you a heads up, we will be expanding the target scope. We have started with the Indoor Ag Science Cafe Indoor Vertical Farm because of the funding. But I think this effort can continue, but having a little bit wider audience. So the entire CEA is going to be the target audience. So it's going to be a lot of fun. But anyways, I'd like to go over some of the upcoming series. So we are doing monthly webinar series to exchange information, science-based information, industries, technologies to showcase. So we have great speakers lined up. February, this is going to be, I'm going to introduce quickly later, but Robert Conrad from Ball Horticultural Company is going to give us a lecture on the seed technology. And then March 5th, international speaker from Japan. We have a slight change in um, usually 11 a.m. Eastern, but this one 10 a.m. Whenever we have Asian speaker, we have to change this way so that they don't have to stay up too late at night. Um, Dr. Shimamura from Hammo is going to share some of the cases, issues of volatile or air quality contaminant, VOC, and how that is impactful if you are not familiar with that issue. April 9th, uh, the scientist in Wageningen University is going to talk about speed bleeding and how far it can be strategically used to speed up even the bleeding process. So that's one of the recent topics of utilizing indoor environment for bleeding. So that's something you want to look forward to. And then May 28th, again, 11 a.m., 3M is going to give a presentation on thin films technology. That's also interesting to change sunlight light quality. So that's May 28th. June, again, from Japan, Denso is interested in talking about, this is logistics optimization. So they are in the group of Toyota Corporation and Toyota methodology, how lean production operation can, make sense in the CEA. Interesting topic. So I invited specifically for them to talk about that. And then there's no cafe in July. So as usual, that's our summertime. So we are not going to do this in July. So that's an upcoming schedule. Schedule will be updated in this OptimIA website event schedule page. So check out if you want to look for what's going on, what is coming up. So also we started advertising more basic knowledge of indoor farming OptimIA University. This is a great series of topics and basic information to cover. I don't think seed technology is included, Robert. So maybe you can you can add that in this university. All right. So I'm so glad we can have Robert Conrad from Ball Horticultural Company talk about seed technologies. He has excellent laboratory to provide these technologies to wide range of horticulture companies and scientists also in academia. And then he's going to give us basic information on how seed technology can contribute and optimize crop performance. So with that, I'm going to stop sharing and then have Robert start sharing and start the presentation.
All right, are we set?
Great.
All right, this is a pretty broad topic, right? CEA and bedding plant production and leafy green. So I wanted to kind of narrow it down that I will be talking about my experience mainly with bedding plant production, plug production, and leafy green CEAs. So just as a start there. I work for the company Ball Horticultural Company. It's a third generation private family-owned company in the US, but it is very international in scope. Transition in our fourth direction. This is all the locations. So we basically have a worldwide presence. We're defined mainly as a breeder and producer of soft color. So that's from bedding plants to by seed to cuttings to tissue culture, roses, but it's a pretty big organization. But part of the organization I work in is the supply chain for flowering ornamentals. So as a company, we are down at the breeder producer level all the way through the distributor level at different markets. Here in West Chicago, I work for the distributor Ball Seed Company. Internationally, the breeder producer that I mainly work with is Pan American Seed, also Keith Seeds. So they will be representing the different markets, but we stay below that finished grower line. So if you look at the bottom right, you'll see seeds and cuttings. That's the main, volume-wise methods that we distribute our genetics through the world. And my part of it is seed. So the type of products that we typically do are, our motto is we want to color the world. So if you would think of basically, things you would buy in the spring to put on your patio. So mainly it's flowers, but it also includes vegetables. So vegetables you would have in pots or you would have in containers. We actually even have fruits that you do in containers, raspberries and actually blueberries that are hanging basket type blueberries. So just different and unique products that we bring to the horticultural market. So my part of this is managing the post-harvest seed technology. So that's pretty self-explanatory. Once seed is harvested, before it is shipped, that's the post-harvest phase, and that falls under areas that I work with. And most of that work is to try to give uniform stands that can be achieved with automation. So I throw this slide up there because it gets kind of confusing. I work for Ball Horticultural Company. The product line that is you will hear most is the Ball Premier line. That is the trade names that all these products are sold on. So like there's a whole lot of internet information about our lab. It would be like if you would Google Ball Premier Lab, that's where you would pop up a bunch of videos and a bunch of different presentations and public information about the labs and processes if you need it for your class or something like that to show a short video of what pelleting looks like. For instance, you could hop on the internet, put in Ball Premier Lab, and things will pop up, the resources you can use. We're pretty open to tours. So most of our information is pretty public. Third-party services like that when we sell these processes to other people, so we'll take in seed and we'll treat it, and we'll coat it, we'll pellet it, whatever we do for it, and it goes back to the customers. That is done under Seed Technology Services, that name. So the two names you'll hear me talking about most, because these are the facilities I'm working with, are the Ball Premier line and Seed Technology Services. About half of actually now it's about 70% of the seed the corporation produces goes through our lab for one form, enhancement, or another. Quite honestly, the number one reason is for coating and pelleting, so that the seed can be sown with high speed with automation. But it's a sizable operation. If you look at the right of this slide, all those dark lines are different species we work with. So there's being an ornamental horticulture company, we have lots and lots and lots of different varieties of plants or species of plants. And within those species, there's lots of varieties. So for instance, if I always talk about on the priming side, we prime lots of pansy seed or viola. There's about 250 different varieties of pansies we prime. So when one of those dark lines on the right, one of those lines would say pansies. So on the left, under the seed technology at Ball, these are all the different trade names you will see in the industry, which are really combinations of technology. The industry is a little different than the traditional vegetable seed industry, where you have raw seed, and you could buy pelleted seed or prime seed or all the different forms. In the bedding plant industry, mainly we bring seed with a package, right? So you can only, for instance, buy pelleted petunia seeds, or you can only, for instance, buy coated marigold seed. We don't offer lots of forms to pick and choose from. We use the technologies to help us achieve quality levels, and it's all rolled in. So you see it sold under different trade names, not different not different processes. And this information, once again, is all online. This comes from the ball seed catalog, which would be online. So it's just literally a catalog page that I pulled out. So we do have two different branches. One is in North America, which is where I say Seed Technology Services. We also have an office in Europe, which is called Seed Tuning. Seed Tuning helps us, we had the same processes, but historically Seed Tuning does more of the perennial treatments. Seed Technology Services does more of the bedding plant treatments. But as we've grown as a company and corporation, you can basically get all the different processes at both locations, which helps with the phytos and having to ship seed around if you use the services. So, all right, so enough of that. Enough of the talk about, who we are and what we do. So now let's get into this. I thought I would start this presentation basically by saying, in your minds, what industry is closest to the vertical farming? So, you can look at whether it's field-grown veggies, transplant veggies, cannabis production, traditional greenhouse operations. I'm going to give you the reasons why I think traditional bedding plant plug production is really the closest industry to the CEA leafy greens industry. So in saying that, let me move this over a bit or minimize it even. So in saying that, the main similarities between the CEA industry, when it's CEA with leafy greens and with bedding plants, is uniform is critical for your seeds for your stand, right? So both crops are blown out. And what I mean by blown out is a seed is sown into a tray, a plug tray typically. The plug trays are grown until they start to overlap. Then so for instance, it could be 2 days, 3 days, 5 days, 7 days, but it's very, very young, very into the growing cycle. Those flats will go on a conveyor belt under a computer typically, and the bad ones will be blown out, right? So they literally use air to empty the cell. Sometimes those cells are refilled, sometimes they're just blown out. So a seed, to be considered a viable seed, has to survive that blowout process. Another day, for instance, and your stand might improve, but the way it works is they all got to be ready at the same time or none of them are ready. And that's true basically in both industries. Growing zones have multiple species in the same zone, right? So when I go into the leafy green type facilities, I got lettuces and kales and brassicas and a whole bunch of species within one environment, and in most cases within one water management system also. That is very, very similar to bedding plant plug production. You can walk in a greenhouse and one bench can have, petunias and ageratum and zinnia and dusty millers and zone after zone after zone. Nothing is dedicated to any one species. It's all that week sowing, and that week sowing could be, 250 different species. And then of those species, like I say, multiple varieties. So that's pretty similar to us, where you can't really optimize production for anything. You have to get things to work in the growing zone. The other thing is the crop morphology. Both of us are really using our cultural practice to change the morphology of the seed. In the bedding plant industry, what we're looking for is a very tight, very compact, budded plug. In the CEA industry, you're looking for leaf expansion and the ability to harvest. So we use PGRs to modify our growth. You guys tend to use light recipes to get different growths. So we're both highly automated and we both do very quick turns. Now, if you look at the differences, there's one huge difference in our industry, that plugs are grown with overhead watering, where leafy greens typically have unlimited moisture that they're given and available to. So the difference mainly is how the product is used, right? So they look almost identical, stage 1, stage 2, and I got some slides that'll show you how we do that, and you'll say, wow, that could be our facility. But our plug is shipped to somebody that will use mechanization to pull that plug out of the cell and put it in a pack that they will put, say, 4 to 5 more weeks on that pack. And then you'll go to your garden center or your big box store, and that is how you'll buy it. So the whole product gets removed from the tray and transplanted, just like when you guys are filling your growing RAS or medias or however you're growing, you're finishing your product. But ours is done at a much later stage, so you have to keep the roots within the tray. So if we grew on, say, an ebb and flood bench or whatever, we would have lots of roots coming out of the bottom of the tray. If the roots are coming out of the bottom of the tray, we can't pull those plugs then and transplant them. In the leafy green industry, the roots and media are literally left behind as waste, right? You're going to cut that off. You're going to take what's growing, you're going to bag it up, and that's the product you sell. And the roots and the media, whether that's a actual pot or actual floating bench or actual, just grown in some other type of media, like a mat, that part becomes waste. So there's a lot of challenges that are similar to both industries. Now, it's for different reasons. Like in our industry, we can't chemically treat seed. So, and when I say treat seed, I'm talking mainly fungicides. So there are legal fungicides for flower seeds, but there are no legal fungicides for flower seeds across markets. And when I say markets, I'm talking about US, North America, and Asia. So for instance, in the US, we could use a product called Thiram to control fungal growth on seeds for, it's labeled for seed application. We can't use Thiram in Europe, it's banned. So the fungicides you can use in Europe, you can't use in the US. The way our industry works, it's us and our competitors basically have one global inventory. So the seed that is sown in, California this time of year is the same as the seed that is sown in Netherlands or Holland or Australia. It all comes from one source, so we can't treat seeds. We don't, like the vegetable seed industry will treat seeds going out the door. We can't do that. For your industry, it's you don't want any chemicals on your seeds. So both seeds are sown in the raw state and you have to deal with pathogens. They're both sown in cells. We sow in cells and you guys sow in cells. So for instance, our main plug tray size is a 288. That 288 tray, the way we water overhead, is every single cell is a unique growing environment. So it's because it's walled in and it's watered overhead, like one cell, which is basically about 1.5 centimeters square, is completely different from the next one. Grading and patching, we're both repairing. We are basically repairing and blowing out the bad plants and putting good plants back in that plug. You guys are grading as you're filling your rafts, for instance. You're taking the bad plugs and just not using them. The scale is actually very similar too. When I go into a bedding plant producer of plugs versus when I go into a large CA facility, the amount of units you do per week and the amount of bench space you have, yours is vertical, ours is horizontal, but the scale is about the same. One huge difference, those plugs have a peak. We're dead in the heart of that peak now. Greenhouses get rid of poinsettias and start bedding plant productions, and so they're in the middle of their bedding plant production season now, which will basically go Mother's Day till 4th of July. So the difference is CA, it's not quite as hectic. You have the same pace roughly every week. So it's week after week of the same thing. But when you add up those units, in our industry and your industry, the scale is pretty similar, which really surprises me, or surprised me when I started to find this out. So I got to get to the right page here. So I'm going to talk about now the main functions of what we do with seed in the Ball Premier Lab. Remember, this is all post-harvest seed production. So if you talk to people and you look at the marketing view of what goes on here, they're going to say we do seed priming, felting, coating, and testing, and it's sold under different trade names. What I'm going to present to you in the next couple of slides is what we do. It's really wet seed treatments, which people consider priming, dry seed treatments, which people consider coating. Pelleting can be used for dry seed treatments, but basically we use the coating for dry seed treatments, and testing. Testing is a critical function of what we do here. So we'll go a little bit more into those areas, but generically it's priming, pelleting, coating, and testing, but that's really, there's a lot more that goes on under the hoods in each one of those stages and what you can do in those stages. So, but people mostly associate seed technology, post-harvest seed technology, with priming. So it's typically the academic starting point for post-harvest seed technology because it's very simple to do at scale. You don't need huge quantities of seed like you do with pelleting research or coating research. you get highly reproducible conditions and highly reproducible results, because you're working with Petri dishes, and we're doing our research the same way, working with Petri dishes and incubators. The environmental conditions are easy to simulate. You can do it, say 500 seeds at a time. Our typical treatment is 500 seeds, and we'll sow 350 seeds for our tests. So for us, though, priming is just one of the tools in the toolbox. We have many different methods of priming even, and the different methods come in for different reasons. So if I look at what's going on in the publications, this is an article cited on the right, but I read it not too long ago as I was getting ready to do this talk, just to see, okay, where do we stand research-wise in priming? I was surprised, like we're starting this curve at 1990 and it goes through 2020. So the curve on the left is the publications that mention seed priming. The curve on the right is the citations. So if you can see, both of those are growing at a steady pace, right? So there's more and more and more seed priming research. More and more and more is being learned and applied. I would have thought when I started to look at this that it would be more stable. To me, a lot of the priming research, I started priming research, for instance, in the 80s, and I just would have assumed, and I incorrectly assumed, that we're basically onto other ways to enhance seed now. But you can see by the literature being published that it's a growing area, and people are using priming more and more to help overcome different limitations in seed stand establishment. So the difference is, obviously that on an academic side, you're doing more basic research, trying to understand what's going on in seed priming. And I'm way over here on the right side, the applied area of applying the seed priming research. And then obviously there's the transitional areas that both of us do, right? So, excuse me, as the different questions come up in the research, how can it actually be applied? So we started the transitional and work towards the applied. So one thing that people don't realize about priming is that priming provides the perfect conditions for seed pathogen increase, right? So we're constantly struggling with this. And the other thing is, if you can soak a seed in a priming solution, I'm putting this out anecdotally, and you'll see why in a minute, but typically you can water plants with it. So the ways that we control our pathogen growth as we're priming seed. We're going to have seed in a tank. That tank's going to have solution. The solution is going to be bubbling around. And in that tank, we can do stuff like put hydrogen peroxide in there. We can put chlorine in there. We can put UV tubes in the priming tanks. We can make our priming solutions with plasma activated water. We can have ozone bubbling in our priming tanks. We can put biocontrols in our priming tanks, anything to help decrease the pathogen increase as we're priming seed. Just to finish the thought there, some of the other things we do in our priming medias is we use salts a lot just to make it inhospitable for the pathogen to increase and to continue. We can also do things like change our solutions or use biocides. So there's lots of things we have to do in commercial priming of seed to control pathogen growth as we do it. And there's no one way to prime seed, right? So just for instance, in our facility, we have 3 different production methods. We'll have a thin layer priming, we'll have what we call a 3D priming, and we'll have bubbling columns. In our Dutch facility, there we will also use drum priming and other ways to prime seed. So everybody has different methods of doing it, but everybody's going to struggle with the same problems of how do you stop disease growth. To me, this is very similar to in your tanks as you're ebb and flood watering or however you're doing it, how do you control the pathogen, right? So these things that you're seeing on the right, we're both doing research on the same thing, and how do you control this pathogen growth amongst a variety number of species and stop it from spreading? So for us, in addition to priming, we use what we call dry seed treaters, right? For us, they're fluid beds, and I'll show you pictures of these in a little bit so you can see what we're talking about. But fluid beds and side vetting pans are what we use here. Spinning disc, I believe, would also fall under this category of dry seed treatments. right? So for us, we're putting 10 to 15% solids on seed. We're spraying seed with solution, and that solution, the solids in that solution is left behind. But before we spray the solids on this solution, we can do stuff like spray peroxides or spray different chlorine-based treatments or spray whatever it's going to take to clean the seed. A lot of times just putting seed in a fluid bed treatment machine and turning it on and bubbling it, you can mechanically remove spores from seeds so that the seed spores are just being removed in the process. So you need dried seed treaters because a lot of crops that we use have a mucilaginous seed coat. So for us, it's salvia and alyssum, for instance. When that seed gets wet, the muc layer comes out and it's very, very difficult to dry down. In the vertical ag industry, it's more like rosemary and basil. Once that seed gets wet, it's the same problem. It's very hard to dry down. So lots of these seeds just don't lend themselves commercially to priming treatments because of the handling problems. And now that you've primed the seed, how do you singulate and sow it? So we turn to dry seed treaters just as much as we turn to priming as methods to apply the technology to get good stands. The difference is, though we're spraying seeds in the dry seed treaters, they're dried instantly, and that'll come up in a little bit later. So if we start down the path of, okay, we're going to look at a crop, and I'm going to end up talking about spinach as one of the crops we've looked at. But if we're going to look at a crop, the first thing we got to do is look at seed testing. Seed testing for us is done in plug trays. It's done in the same environments that we will use commercially to our customers who will grow these crops. So this is just a picture of a lady sowing trays. You can see the first one here at the bottom right. You can see the seeds in it. She's going to use a vacuum head. She's going to make sure there's one seed per tray. So this is where I say, this is our testing conditions. Our testing conditions, we always set up so they mimic commercial conditions. So this is stage one. We'll start in a fog chamber. Stage two will be a vertical bench, interestingly enough. Now, ours is set up for overhead watering, but so we try to mimic commercial conditions. So this basically is commercial conditions for vertical farming. That's one of the reasons there's such a tight alignment and tight benefit that, that we can work together across the industries and help solve problems. All our plugs are image. We use image analysis in basically everything we do. One of my guys, Shelley Hughes, he does imaging for us and writes our script and writes our codes. So you'll see on this line, there's 4 laser lines basically under that gray cord to the right. In our industry, if a seed germinates and does not become used as a part of your final product, it is typically because it is either too small or too short. So in this system, we're using lasers. Those red lines we're triangulating. 2 of them read leaf area. 2 of them read the height of a seedling. So as we run the seedling under this testing system, we're getting leaf area and we're getting height, and then that black box on the left that the camera's showing through gives us a color image just for reference. But the data we get from our seed testing comes from those 2 lines to the right. We do the same thing with Petri dish testing. Our lab testing is automated. And when I say this, we're not 100% automated because we do so many different species that they're not all compatible. But, it's probably up to 70% of everything we do, we're reading with the computer. So this is an example of how we do, typical, laboratory type testings. We throw that red grid on there. That red grid is mainly a reference. It makes it easier for our people to see once they sow seed. Is there a seed where it should be? That is just a sandwich box or a typical germination type box used in our industry for germination testing. On the right side is after it goes in and out, there's a laser line that catches it. So we don't read seed based on when the root comes out. Our systems are looking for chlorophyll. So we're looking we're defining germination beyond the root emergence stage. We define it when chlorophyll appears. And so most of the times you first see this, you can't even see the green chlorophyll, but it's there. And we do this because roots can be very tricky for us, particularly from an imaging standpoint. At what point in time do you say the root has caused germination? So our testing is really to show small differences in treatments So we need a very uniform point of germination. Think of a pepper, think of it, as the pepper imbibes, for instance, the root can be pushed out and hasn't even started growing. The imaging system can pick that up as a root. Same thing with flower seeds. There's just a lot of different ways to manipulate root and root length and root growth. So we just switched to all our testing. You can see on the right, that's picking up chlorophyll signals from the seed. So whether it's plug testing or petri dish testing, it's automated. And we've taken this automation also to final product testing, for instance, with pellets. The thing on the left is there's a box that we'll pull out. We'll just throw a random number of seeds on and we'll slide it under there. So each one of those green squares is identified a pelleted seed. And if you look at the computer screen to the right, you'll see that histogram. That tells you how uniform the seed of the pelleting run is. It's very important for us to have the same size and shape with our pellets because typically our product, a lot of the time, like 30–40% of the time, in flowers is sold as a mix. When you go to the garden center, you usually don't buy a straight color of a pansy or a straight color of a petunia. You will buy multiple colors in that pack that you use. Or if you buy a basket, think about, that's used from single colors, but we don't pellet seed as a mix. We make mixes after we pellet seed. So it's very important for us to have everything the same size and shape. And that is also very important for our growers because as they're going, like I said, petunias, 250 plus varieties are pelleted. They're sowing 250 plus varieties every week. They don't want to set up their watering line and their sowing machine different from every pellet. They want to just be able to fill the hopper and away they go. So now we'll talk, I told you I'd show you some pictures of fluid beds. These are our dry seed treatment facility. So on the left-hand side, you just have an overall shot of the room. There are columns there. And like I say, if you go on the internet, there's actually, you can see pictures of these seeds running. I didn't want to bog down the talk with stuff that's very easily available, but there are columns over there. And the center picture shows what one of those columns look like. This is an 18-inch column. We have everything from 4-inch to 18-inch columns. So we have a lot of different sizes because flower seeds come in a lot of different sizes and a lot of different volumes. But what it does is those are nozzles on the bottom of that column, and they're shooting through. On the far right, you can see there's yellow seed in the column, and you'll see a Cole Palmer pump there on the bottom right. So we pump solution into these columns. It is mixed with warm air. We run our intake air at 45 degrees Celsius, and our exhaust air, because we're getting evaporative cooling, is usually about 30 degrees. So we can continuously spray onto seed, and it ends up coated, which is typically like that crop blowing around there is probably marigolds. And marigolds will have 100% buildup on the seed. But before we start building up the seed, we can take whatever solutions we want and spray the seed. So whether that's a bleach solution, whether that's a peroxide solution, whether that's an HA solution, whether that's an enzyme solution, whether that's a PGR solution, for instance, if it needs a growth regulator like GA on it, we can spray that right on the seed before we apply the final coating. And the seed never gets wet. I mean, it's wet as it's sprayed, but it dries as it goes. So these are typical fluid bed dryers. I mean, if you talk about the whole name of them, the industry would call them a fluid bed dryer because the way the product flows and dries. So this would be a typical product. The top left would be raw seed. The top right, and this is marigold seed, the top right would be a coated seed. That's the base coat. The bottom left is the finished product, and that is a very, we have done that so it goes from a product that doesn't flow well to a product that does flow well. One of the commercial products we sell is something called ball control growth. We have a chemical, it's acetamidol, that we can put on the seed to stop stem elongation. And when we do that, the product is orange. But it's a very, very simple way for us to expand what we can do just in the priming columns. So everything you can do in, not everything, but a lot of things you can do in priming columns, you can also do in these fluid beds. A lot of things that you can't do in priming that you can, so there's different ways that seed can go in and out of conditions. Remember early on, I said, we don't really sell we don't have a pelleted line and a coated line. We just have an enhanced product. right? So marigold, for instance, you don't buy raw marigold seed, you buy coated marigold seed, and that's just the way it comes. And that could be treated for something to do pathogen reduction underneath. It could be growth regulators. It could be whatever is needed applied to that seed in the production process. So this is our pelleting room. I assume most of you guys people know pelleting. Just like wet seed treatments, everybody associates with priming. Dry seed treatments, everybody associates with pelleting. This is the reason most of the seed runs for the lab, quite honestly, because you take petunia seeds, are 250,000 seeds in an ounce. Begonia seeds are 2 million seeds in an ounce. They're finer than sand, and they're all built up to be able to be sown high speed with precision, basically around 700 trays an hour. So we have a dozen pelleting pans in our pelleting room. Most of our pelleting is automated. Once again, this is something we do internally. You can see on the left side, there's a spray nozzle, there's seed in a pan, and there's that black tube shining the light. That black tube shining the light is an NIR meter that is reading the moisture of the seed as it's flowing around in the pan there. On the far right side, you can see the program, once again, done in-house. It's driving the liquids and the powders that are being applied to seed. So it's reading a recipe for that crop of that quantity and size and applying it to the seed. So we also do manual pelleting, but for instance, 70% of the volume of pelleting that goes through our facility is done automated in 2 pans, and the other 30% is done manually with 8 pelleters. So because we have so many small batches and oddball batches, we can't go 100% automated, but by volume, most of the stuff we do is automated. This is a familiar machine. If you strip away all our stuff, that's a spinning disc. This is very, very commonly used in the vegetable seed industry. This would be applying what they would call a film coat. We can use this also. That same black tube is on front of it. We automate the spinning disc, so we can actually do pelleting in there, or we can do biological applications in there. So it's just another one of the tools in our toolboxes. We take seeds to whatever equipment makes sense, not necessarily worried about the product line definition. So one of the things we do a whole lot of is multi-seed pellets. In the flower seed industry, the seed quality isn't always the best. The number one reason people are buying bedding plants is because of the way they look in their garden. That is not necessarily in line with how the seed's going to germinate and perform. So a fast answer for seed quality issues that helps seed get to the market fast is a multi-seed pellet. So on the left, those are called precision multi-seeds. Those are done in a machine. I'm going to show you in a couple slides real quick how we do it. On the right is just regular multi-pellet. Those are actually done in those pelleting pan by those 8 pelleters I talked about. So they are sticking different combinations of seed together into one unit, and we will here do the testing of that unit. So basically you can take, something that germinates at 70% germ, you take, 3 seeds, and now you just by the numbers, you're going to be at 85% germ. And we would do that quality control in-house basically. So when people sow a multi-pelleted product, they know what they're going to get. So the way we do that is we actually coat and pellet the seeds ourselves and run through commercial seeding lines. So these are just commercial drum seeders where we've pelleted the seed, but we don't go into a plug tray. If you see between those 2 drums there's orange seed and there's yellow seed, and in between you see that white tray. That is a very dense setup. That's every ½ inch or every 12 millimeters wide. So in that tray right there in front of you, there's 400 different cells that are going to be sown with seed. The white tray is basically a moving funnel to direct the seed. This is what's underneath that white part. That top hole is 1/16 of an inch, I believe, maybe 1/8. There's different size trays for different size products, but you can see the individual seed, the exact mix of individual seed we want is in each cell. And you can see where the seed is. It looks like it's a little wet, almost filled with water. That's actually a water-soluble type wax that we injection mold in there. So we melt it. Nozzles come down after it's sown. We spray the liquid into there, and then we come up with that product they were doing today, this day, when I took the picture last week, was tribasil. It's a product we sell a lot of. That's what the actual seed looks like. So when you sow the seed into a pot, one seed unit per pot gives you that mix on the right. That's what we call tribasil. That's one of our top selling multi-seed basils. So we do this to basically, the customers these will be the pots that you buy in a Home Depot or a Menards or a Canadian Tire, or, the different big box sellers in Europe. You will buy one seed, it turns into a plant like that. We do the same thing with salads. These are different mixes. Specifically, we'll put together different salad seeds to give this combination. And I don't know, I think there's about 8 or 10 of them that the breeder producers and marketing staff and product managers put together and say, these would be good mixes. We just put the seeds together, make them, and get them to germinate. It's entirely another part of our business to determine what to put together into these different mixes. But I wanted to give you an idea of some of the technologies that are being used in our industry that could or could not be used to your industry. So this is an interesting crop. It's Emerald Falls Silver Dichondra. And really, this crop is the reason I'm giving this talk today in a roundabout way. This is a product for us that is very interesting seed physiology. It is dormant. I mean, it needs to be scarified. It won't absorb moisture, right? But it's not because it's a hard seed, it's because it's a waxy layer. So under that seed, you have to scratch through the waxy layer so the seed will imbibe. Interestingly, this is kind of a fascinating crop if you're into seed physiology, because interestingly, is if you remove the seed coat, this thing goes from germinating in the light or the dark to be an absolute requirement that it needs dark. So it's a difficult crop to scarify. It's a very popular crop for us. It's usually not seen in this form. Usually it is, if you look at pots, the way pots are put together, hanging baskets in particular, you have what they call a thrower, the main focal plant, a filler, something to take up space in the pot, and a spiller, something to grow over the edge of the pot. This would be the filler in the pot. So this would trail down as you grow. So we developed a seed technology in the last couple of years to be able to scarify the seed better. We moved from a mechanical scarification to another type of scarification that was able to overcome our problems in trying to get seed to the market. So we were constantly short on the seed because we didn't have good scarification technology. This scarification technology works for spinach. So we took a look at, Cherry actually wanted us to take a look at spinach and see if there was anything we could do to get it to germinate well. So this is, I'm not going to talk varieties or genetics or companies or anything like that, but this is 5 different lots of spinach that we got from one of the breeder producers to take a look at and try to do this with. And so in all our work, the first thing we do is we do a seed test, right? So we have to understand exactly our starting point. And interestingly, every one of these packages of seed came to us with a 90% plus germination rate. So the blue lines there are what we get in our plug truss, and I'll show you the plug trays a little bit later. I mean, it's the next couple of slides, but how we grow the seed, we got anywhere between 37% germ and 71% germ. So you can see that third lot is the best lot, and then you get to that far right lot, and it's just not a good lot, either raw or treated. But you guys as growers and us as customers of these companies from a distributor standpoint, the information we get on this seed when we receive it in our hands is it's 90% plus germ. So that's one of the reasons we always start with those germ tests, and one of the reasons everything's done with imaging is to be very standard and very particular in the way we do our testing. So these are those same spinach lots. I'm not going to go over every single lot, but this is, the first variety. The raw seed is on the left. The treated seed is on the right. So what we've done with the treating it is we've reduced the pathogens on the seed, right, through different ways of, definitely the methods I've just talked about is one of those ways to reduce the pathogen growth on the seed. We've done that, and then we've cracked the pericarp. In this case, it's pericarp cracking because when you germinate it under high moisture conditions, the seed needs more oxygen. So the same thing we're able to do for the dichondra we're doing with the spinach. So this is another lot. You can see, like, if you just put raw seed on a tray versus this treated seed on a tray, you can see the differences. And like I say, when you're in an individual container like this, and it all has to be the same, you can't wait for 2 or 3 days, weeks, months, whatever time frame you want to use. I'm sure that lot on the left would eventually get to 90% germ, like the label says, but not in a 90% usable manner for anybody in our industry. And this is relatively new for the breeder producers of spinach. Like, who's ever heard of putting spinach in a plug tray, right? Typically, this is sown directly in the field. It's not a transplant product. And in the field, you don't have that very, very high moisture that gives you these very specific germination issues. So this would be the same thing, the same lots, single seed at 7 days. So when you take a look at just these treatments you can do on the lots of seed, these are the same five varieties or 5 lots. This is the first one, non-treated on the left, treated on the right. And so this is not a wet seed treatment too, right? This is not seed priming. So, this is a good example because I keep talking about these dry seed treatments. This is totally a dry seed treatment here. So this would be the next lots and the next lots. And, whether it's you hold these differences all the way through between the treated and the raw. This is the way we're growing these, are ebb and flood benches. It's very similar to the way we do other crops, except in this case, we're bottom watering. As we evaluate varieties and evaluate genetics to see how they do in this system, once again, just like with our plug testing that I showed you pictures of, we want a very uniform system to be able to judge things. So those same 5 variety lots, the non-treated on the left, the treated on the right, we took 1 plat of each and grew them out for 30 days to see what the difference would be just to this dry seed treatment. So this is what they would look like 14 days after the fact. This I put this shot on the left because it just shows you our lighting setup. It's red and blue. In this case, it's Sylvania strips with white LED lights. DOIs are around 12. And when we're doing this test, we're doing purely a biomass test, right? So we're not saying the quality of our growth. We're just trying to get a good way to test seed and test these seed treatments on how it's going to affect your guys' yield 30 days later, for instance. So this is just biomass of those 5 varieties, how much biomass you can gain. In this particular case, was 27 days after sowing. So these different treatments you're doing from the get-go to give you a more uniform stand, they're showing all the way through in the yield, which is typical of our industry too, right? Because one of the things I started with is saying how fast our turns were. So that's basically the presentation I have, but I just wanted to thank you for you guys' time and interest and give you an idea of what we do and how we do it.
All right. Well, thank you so much, Robert. I really appreciate sharing the basics and then also the most recent trials and technology development you are doing with spinach.